Hello, welcome for those of you that are here. Right, for the next around hour and 45 minutes, we will be doing um, various science things. Now, if you have any specific requests, I will try and get to them. But what I'm going to try and do is pick out a couple of, well, maybe one each from each of the three sciences. A topic that generally I find that people don't do that well and things that are quite commonly asked. For example, I may start with this. There is a Q&A thing, by the way, if anybody wants to ask stuff as a chat. I think I can see it all. So please feel free to say what you like. Right, I am going to just go straight into it. I'm going to talk about stuff to do with the heart and the lungs first in biology and the kind of important parts of it that commonly get asked and that I think you should be aware of. I think then I will move on to a chemistry topic, part largely to do with two things. I think electrolysis is often something not done very well, and quantitative chemistry. I want to just talk a little bit about that. And then in physics, I'll see what I feel like. If you are doing triple science, then I will try to avoid some of the triple science topics today and then try and cover more of them tomorrow. But everything else is still relevant to everybody anyway, so that's what I will do now. Um, right. Also, just as a separate point, for those of you that are usually with us in terms of our students, um, the usual thing that we do on a Wednesday is still happening. So you may be here quite a lot. So if you want to join that, you are welcome to later on. Right. So circulatory system. Let me just see what I've got in the. Make sure I've. Okay. I'm going to answer that question later. Right. Let's get into the circulatory system stuff. So we have got. Oh, you can't see my screen. Um, can somebody else just message and say that they can see my screen? Um, I'll just stop this and start it again. Occasionally that is an issue. We can, yeah, fine. Okay, right. I'm gonna have to carry on, I'm afraid. There might be an issue with your Zoom. You might need to just click around a little bit and get it. But my screen is being shared, and you should be able to see something that says the circulatory system on the front. Right. Let's go into these. These notes, by the way, are technically anybody can have, so you know, technically. But if you're not sure how, then ask. But that doesn't apply to all of the notes that we use, just to these biology ones for now. Well, I mean, the answer to the question about is this biology paper one or two, or is it all of the sciences? The um the the answer is I'm just doing anything really for this one. This is more generic. Just I've opened it up to everybody. Anybody can join this, and I'm just doing some fairly random topics. So I've just picked this one out. This in particular, if you're doing AQA, is on paper one. I don't think that really matters too much. But I will be doing a biology topic, the quick one. I will do a chemistry topic like electrolysis or something, and then a physics topic, maybe just the energy one. I don't know. I haven't really decided. I'll just decide. But if you have any specific questions on specific topics or anything like that, then you can just ask. Can we see all questions of other students or not? I have turned off the feature for you to be able to see the questions of other students. I think just because I think it's better that way. Right. Aerobic respiration is a process that happens in all living things. And when you think of the word respiration, if you don't think immediately of the word energy, then you will probably lose some marks. Um, Topic four, chemical changes chemistry, will work. Yes, because that's partly electrolysis, so I will go through some of that. So if you talk about respiration, you should immediately think of energy. The other things that associate with the word respiration, just I'm speaking on the basis of what mark schemes often have. mitochondria, because that is where in a cell the process of respiration takes place. And the process of respiration releases energy. So often these things combine in forming like several marks for an answer. Like more mitochondria means more respiration, means more energy released. Try and combine those, your understanding of those different concepts, right? So this is the equation for aerobic respiration. It requires glucose and oxygen to happen, which means that if you had less oxygen, then you would be able to do this process less, which means that you would get less energy, right? These kind of processes all link together. And understanding the equation helps to make sense of lots of topics where you have to apply your understanding. A typical example would be why, for example, um, this is a very simplified example, but for example, if somebody who smokes, why do they find it difficult to exercise? Then the way you would begin to form an answer to that question is by explaining that they would, for various reasons, get less oxygen around their body. And if they get less oxygen around their body, it means their cells will do less of this process called respiration because it requires oxygen. If they do less respiration, they will release less energy. Therefore, they will find it difficult to exercise. You see how we've turned that into a 3, 4 mark explanation fully in terms of why it's one thing smoking leads to difficulty exercising. It's by linking all these things together. That's why this understanding of this topic is particularly important. This is the symbol equation for aerobic respiration. You wouldn't need to, I'm very unlikely that you'll need to write out the whole thing ever, but you would often have to fill in a gap. So in essence, you still need to know all of it. Whilst we're not focusing on it today, photosynthesis is this equation the other way around. Okay. Carbon dioxide plus water, arrow, glucose plus oxygen. Is that the other way around? Let's continue. Right. The idea of the circulatory system is that because we need to get energy or release energy, we need the two things that allow us to release energy, which are glucose and oxygen. We have to get glucose and oxygen all around our body to all of our cells so that they can do this thing called respiration, which helps them release energy. Um, ooh. Um, I got this question on my mock. Explain the effect of a partly blocked artery on the human body. Um, yeah, so I will answer that question because we will get to it in the perfect question to be able to explain what we're doing. So we need to get glucose and oxygen all around our body to all of our cells so that they can do this thing called respiration to release energy. You see what I mean? You've got all of that is important. Now, initially, we will focus on the oxygen. For the part of this topic, the majority of thing that we focus on is the oxygen and how the oxygen goes all over our body. The glucose technically is less interesting because we can store glucose. When you eat some food, the glucose gets stored, which means that we kind of always have it. Whereas oxygen, as you know, we get it as and when we need it. If you are sitting doing nothing, you will breathe normally. If you need to get up and run around, you start breathing more because you need to get more oxygen around your body so that your cells can release more energy to keep you running. So that's why the oxygen is a little bit more interesting because it is constantly happening, whereas the glucose is stored. Um, so the way in which we transport oxygen around our body is mainly due to the lungs and the heart. And the lungs and the heart and the blood all work together to ensure that our cells get what they need to be able to release energy from respiration. So in order to get energy, we need to get oxygen all around our body, and it's the lungs, the heart, and the blood in conjunction together that work to get that around our body. Let's go straight to the lungs. And again, people who I've taught will have seen this a million times, but I want to tell you anyway, which is that imagine this is oxygen in the air that you are about to breathe in. What we need to understand is why we're breathing this oxygen in. We're breathing the oxygen in so that we can get it all around our body to all our cells so that they can do this thing called respiration. When I often ask a question that says, why do we need oxygen? And people say, to breathe or to live. These are very vague answers. You want to be very precise about it and say, well, the oxygen is used in the process of respiration. Thank you. Thanks. It's used in the process of respiration. So what we then want to do is understand where the oxygen goes next when we're doing all of these things. So here is the oxygen. First, we breathe it in through our nose and mouth. It goes down our trachea. That should not be labeled. And it goes into a lung. And there is a lung, and it's the same on both sides. Just I've drawn half of it. The oxygen moves through these things called bronchioles into the alveoli, which are these little air sacs, and there's millions and millions of them, like broccoli, at the end of our lungs. Millions and millions of them. Now, so the oxygen just fills them up like little balloons and they inflate slightly. From there, the oxygen needs to move into the blood because that's the blood is ultimately the thing that's going to do most of the carrying of this oxygen around our body. So whilst you can't see it here, there are lots of blood vessels traveling around here, and what they will do is take on the oxygen. The oxygen will move from here into here. If there was a way to zoom in on one of those alveoli, then it would look like this. There is one of them. So here is one of those alveoli that has just filled up with the oxygen that you just breathed in. And from there, it moves out of the alveoli into your blood. So this is the blood flowing around the outside. By a process that hopefully you are aware called diffusion, which is the movement of particles from an area of high to an area of low concentration. It moves from a high to a low concentration into the blood. So that's what's happened. That's where the oxygen has gone. It's gone into the blood. Now, and we're going to continue this in a moment. We're going to continue this line and continue to see where the oxygen goes. But what I like to do at this point is mention other things that you should know. Like you could easily get a question asking you about the adaptations of the alveoli, or describe how the alveoli is adapted for gas exchange, or describe 3 features of the alveoli which help speed up diffusion, or something like vague like that. Something about these alveoli that make them really good for their job. And their job is to get oxygen from here into the blood as quickly as possible. We want this process to happen, this movement of oxygen into the blood to happen as fast as possible. And the alveoli have a couple of 3 features, basically, that are really, really good. I'm just going to mention those 3 features quickly. Those 3 features are thin walls. By having really thin walls, it means that the oxygen doesn't have far to diffuse. We have a short distance for diffusion. If you had a really thick wall and you have to get across it, it takes a lot longer than if you have a really thin wall and it goes across it. So it speeds up the diffusion. The other thing that they have is a large surface area. Having a large surface area is great because what it does is it allows all of these alveoli. That's what makes it have a large surface area. By having so many of them, it literally increases the overall surface area. And by having so many of them, it means we get loads of oxygen going into the blood all at the same time. So again, it maximizes the diffusion. We see a picture here of one of the alveoli, but if there were millions of them, then all of this is happening at the same time. So this oxygen is moving into the blood all at once across all of these other millions of alveoli. So lots of it's happening at the same time. That means lots of oxygen is going into the blood at once, which means lots of oxygen can get around our body quickly. That is the benefit of this kind of thing. The other thing, I'm going to mention it now, which without going into detail, another good feature of the alveoli is that they have a good blood supply. Okay, I'm not going to go into detail about that one. But if you just mention it, it's usually worth a mark. If you have to explain it, you have to say a little bit more. Right, let's talk about where we were. The oxygen that we had just breathed in has gone from these alveoli into our blood. Specifically, and we'll talk about this later, the red blood cells. So the red blood cells are actually the part of the blood that are carrying the oxygen. But for now, let's not worry about that. We'll talk about it later. So let's just focus, let's call it the blood. The blood is carrying oxygen. Now, what I'm going to do is continue this path that this blood is taking. Okay. Remember, this blood contains all of that oxygen that we breathed in earlier in the diagram that I showed you. Okay, so we've taken it all in. So all of that oxygen is being carried by this blood. Okay. And at this point, for obvious reasons, we describe it as oxygenated blood. Oxygenated blood. Okay. Because it's carrying oxygen. So again, I'm going to keep saying this because I think it's really important. The reason we are learning about this is to understand that the oxygen that you breathe in, the one that's the specks of oxygen that in the room that you're in right now, they need to get into your body so that they can go to all of the cells in your body from your head to toe. That oxygen has to go all around your body because those cells need it to do that process called respiration that we mentioned at the beginning. I know that I feel like I might be repeating myself loads, but I can't tell you how important it is to understand that. Okay. The oxygen has to get around your body for respiration. Oxygen is not for breathing. The breathing process was just you getting it in. That doesn't explain why you need it. You just get it in. Breathing is just like sucking it in. But what's for? What's the reason that we have it? It's so that it can get around your body for respiration. Okay, so we have breathed it in. It's gone from our lungs into our blood. Now, where does the blood go next? The answer is on the screen. The blood will head towards the heart first. It doesn't immediately just go all over your body. The heart is like the I don't know what I was going to say there, like a train commander. It's just directing where to go, basically. So we have to go into the heart first, from where it will be sent all around the body. So we have, make that bigger, a picture of the heart. From here, the oxygenated blood, remember this is carrying oxygen, goes into the heart. However, the heart is a complicated diagram, which understandably can make you confused. I like to simplify the diagram a little bit. First of all, the heart is just made up of four sections. Okay, we have something called the atriums on the top, left and right. So left atrium, right atrium, and then we have ventricles on the bottom, left and right. Your left and right are the other way around as you look at them. Right, that gets rid of these labels here. Left atrium, right atrium, left ventricle, right ventricle. Fine, understood that. Then it's about the thing that most people, I believe, struggle with is these names and the names of these different blood vessels. So again, I would like to try and simplify this for you. The heart has got two entrances and two exits. The exits are here. Exit one, exit two. And it's got two entrances. Entrance one, entrance two. Okay. There are two entrances and two exits. So two ways in and two ways out. Now, and obviously the arrows here help, but not every diagram of the heart will have these arrows. So this blood that is now currently on its way to the heart has to come into the heart via one of the two entrances, either this one or this one. Okay. Now, in this case, it's going to come in this way. Okay. One way to try and remember this is, or at least I'm not sure if it helps you remember, but it helps you understand it. This entrance is called the pulmonary vein. So that's the blood vessel that's bringing the oxygenated blood from the lungs to the heart. Now, a quick, easy way to try and remember some of this, which we're going to come to a bit later on, but we have these things called arteries and we have these things called veins. Arteries always carry blood away from the heart. So they're a bit like the exits, right? So these two here that I said were exits have to be arteries because they are carrying blood away from the heart. The A in artery helps you remember that an artery carries blood away from the heart. The veins carry blood towards the heart or into the heart. That's how you can remember it. The veins has the word in. So a vein carries blood in or towards the heart, which means that these two green arrows that I've just drawn must be veins because they're going into the heart. So the middle things, which are exits, are going away. They are arteries. The other ones are veins. They're going into the heart. So immediately, that helps you to understand why this is called a pulmonary vein, because it is a vein carrying blood into the heart. And on the other side, the other entrance to the heart, carrying blood into the heart, is called a vena cava, which begins with a V. Again, it is a vein. It's like a Latin something or other. So that is carrying blood into the heart. That's why it's called this. I just want to make you understand why these namings, why the names are what they are. So a vein is carrying blood into the heart. Another vein is carrying blood into the heart. The arteries are carrying blood away from the heart. So A for artery, and another A called the aorta, again, Latin stuff, is carrying blood away from the heart. That is the aorta. So you can begin to isolate the naming of these by understanding the A for away and the vein in by doing that. Right, next thing that you can understand is what the word pulmonary means. If get rid of this, if you looked up the word pulmonary in the dictionary, you would it would literally say, because I've done this, it would say related to the lungs. The word pulmonary means it's related to the lungs. Pulmonary diseases, for example, are diseases of the lungs. And so, if we put two and two together here, if I've got something called the pulmonary vein, I should be able to now understand, just from the name of it, what it does. It is a blood vessel, it is a vein, which is carrying blood into the heart. Okay, so obviously we're getting blood into the heart, but where is it coming from? If it's got the word pulmonary in it, it's coming from the lungs. So if I ever asked a question saying, which blood vessel carries blood from the lungs towards the heart? I immediately can go, well, towards the heart, it's got to be a vein because it's going into the heart. And from the lungs means it's a pulmonary something. So pulmonary vein is how you can understand that that is what's bringing blood into the heart from the lungs, because that's where we're coming from, right? We're coming from the lungs here into the heart. That's why it's called the pulmonary vein. Right. Let's continue. Again, we have oxygenated blood coming into the heart through the pulmonary vein. This blood, quite hard to see this. I'm going to try and make this thicker. This blood goes into this section of the heart, the left atrium. It is squeezed down into the left ventricle. It is then pumped out this way via the aorta. The aorta, again, begins with A, carrying blood away from the heart. That's all that we're doing. So far, we've only used that side of the heart. That's the only side of the heart that gets used at this particular point. And so this now would continue, and it would now go all around the body. Remember, the purpose of this was to get this oxygen that is being carried by this blood all the way around the body to all our cells. Okay, so this blood will now go to its end destination, which is to the cells all over your body, muscle cells, brain cells, wherever it may be, and deliver the oxygen to those cells. Let's see that actually happen. So if we continued this blood, okay, if we continued the journey, imagine it's continuing. Now remember, we started at the lungs. We just do this. Started at the lungs. Went to the heart, pumped out of the heart. Now we're going to go to the cells in your body. It's going to be all of them, but let's pick an example. Here we go. If I continued this line, it would go to these body cells. Now, these could be anything. Remember, they could be muscles, they could be brain, whatever. But at this point now, the oxygen that is being carried, that we breathed in ages ago, will now move from the blood into those cells because that was where it wanted to get to. We breathed it in so that it could get to those cells, and specifically which part of the cells? The mitochondria, which will use that oxygen in the process of respiration to release energy for that cell, whether it's your brain cell or your muscle cell, whatever it may be, it will do that to release energy for that cell. And that is the full journey that the oxygen takes from up here, right? Where you breathed it in, it went through your lungs, into your alveoli, into your blood. It went to one side of your heart. It was pumped by one side of your heart all around your body and was delivered to those cells. And that is all the journey for oxygen. Unfortunately, the journey, though, is not finished because, as we saw from the equation for respiration, on the right-hand side, we have this stuff called carbon dioxide, which your cells are now producing because they're doing the respiration. So the product of respiration, like carbon dioxide, is in those cells as well, but they don't want it. They want to get rid of the product of respiration. We want to get rid of this because it's a waste. We don't want the carbon dioxide. And we know how we get rid of carbon dioxide because you're doing it right now. You will be breathing it out. And this is how we explain this. So the CO2 now, which has filled up here because it's being produced all the time while we're doing this process, will now move into the blood the opposite way. So the oxygen diffused from a high to a low concentration into our cells here. And meanwhile, the carbon dioxide, which is being produced in the cells, moves the other way. It goes from a high to a low concentration into our blood. And now we have some blood, which we describe as deoxygenated blood, as opposed to oxygenated blood, because it doesn't have oxygen anymore. The oxygen has been deposited away. Now what? This will now need to continue its journey. So this is where we begin to understand why it's called a circulatory system, because we don't do everything in reverse now. So this deoxygenated blood will return in the opposite direction and go back to the heart. However, this time it will enter the other side of the heart. So we used the left side of the heart so far. We're now going to use the right side of the heart. And we're going to enter this side. Again, this has to be an entrance. It's called the vena cava. It's a vein. Veins carry blood into the heart. So it has to be that, right? So we're going to go into there. Try and make this a bit thicker as well. So this is the deoxygenated blood. It goes into this side of the heart. And it's pumped out of that side of the heart. Now let's look at the name of that blood vessel from which it's going out. It is called a pulmonary artery. What does that tell us about it? An artery carries blood away from the heart because it begins with A, away from the heart. And it is called the pulmonary artery. What do we say the word pulmonary means? It means it's related to the lungs. So if this blood vessel is carrying blood away from the heart, and it's called the pulmonary artery, where must that blood now be going? The answer is it must be going to the lungs because it's called the pulmonary artery, away from the heart towards the lungs. And then it's going to be pumped out, and then I can finish this diagram by taking it all the way back to your lungs, at which point you can see that we had carbon dioxide here going the other way. So that carbon dioxide that this is bringing back from those cells goes into your lungs, and then you breathe it out. Meanwhile, the oxygen that you breathe in starts again. The whole thing starts again because you have now been all the way around your body. This whole picture, actually, if you zoom out of it, shows the circulatory system, essentially, because we have the lungs at the very top, we have the heart in the middle, and then we have the rest of the body. So all of your cells that the oxygen is being delivered to at the end of it. And you can see this sort of circulatory system. And you want to really, in a perfect world, be able to recreate this. So what I mean by that is do the following. Let me go down here. Lungs, heart, rest of the body. Okay, so this is the kind of thing that I always encourage students to, if you can do this, it means you get this really well. So we breathe in oxygen, it goes into your lungs, and then your lungs pump that oxygenated blood to your heart. Okay, the blood vessel that goes towards the heart from the lungs is called the pulmonary vein because it's the pulmonary from the lungs, vein into the heart. The heart then pumps that blood out towards the rest of the body, and it pumps out through the blood vessel called the aorta. Right, at this point, all of the oxygen goes to all of your cells, and carbon dioxide comes out. Okay, and that blood then takes the carbon dioxide back to the heart. It goes back into the heart through something called the vena cava, and then the heart pumps it back out to the lungs through something called the pulmonary artery, and the whole thing starts again. So that's a very simplified diagram of a more complicated diagram that was here. So being able to understand that path that the whole blood takes will be very important. Will this be available to rewatch? Yes, it will. It will be on hopefully on our YouTube thing. I think this morning I messed up a little bit, but that'll be there eventually. This one will definitely be there straight after the thing on our YouTube. Right. Any issues, just message us. I'll send it to you. It's fine. Right. The next bit that I haven't entirely talked about, which is other aspects of this sort of diagram. So, for example, I've mentioned these words like arteries and veins and stuff like that. But do we know the actual differences between those? So, there is a general phrase called blood vessels. Let me just check what's going on here. The um, the session is going to be on it afterwards. So the one that I did this morning, just so you know, isn't there yet. It will probably be later on today or tomorrow. But this one hopefully will be there straight away because I didn't press a button when I started recording, but I can stick it on later, so it's fine. Right. So the three blood vessels. I think of blood vessels a bit like pipes that are carrying blood all around your body. And there's three different types of pipe that you need to be familiar with. I've already mentioned two of them, arteries and veins. Okay. The third one is called a capillary, but let's talk about arteries first. We've already established that because it begins with A, um, it carries blood away from the heart. Um, and because it's carrying blood away from the heart, it is carrying blood just as it's been pumped. Okay, so the actual pump at that point, that's when it's going away from the heart. Now, at that point, the blood is at quite a high pressure. And so arch and high pressure basically means it's coming out fast, like it's gushing out, if you want to think of it like that. And so there's a chance that if your pipe isn't strong enough, because it's coming out with such force, the blood, it could like break something. And so you need to make sure that your blood vessels have sufficient protection against that. So they need to have thick, strong, elastic walls so they can deal with that high pressure. That is important. Okay. So if you have a question about, features of arteries or something like that, then you need to explain that they've got strong, elastic walls which can withstand the high blood pressure. You need to mention those things when you do an exam question on this kind of topic. Um, yeah, that's the main thing. So they carry blood away and they've got this thick elastic walls. And also you might want to mention they've got a small lumen. A lumen is the gap through which the blood flows. Which takes us on to, well, the issue with an artery. Why can't we just use an artery throughout the circulatory system? Well, it's because if the blood is carrying oxygen, which it does, as we just showed you a minute ago, the oxygen at various points, like when it reaches your cells or your lungs, has to diffuse from one way to the other. So when it reaches your cells, that oxygen has to diffuse out of the blood into the cells. Now, if the walls are really thick, that process is going to be very slow. It's going to take a long time to diffuse across those thick walls. And so whenever diffusion needs to take place, arteries are not that great. They're great for protection and for dealing with that high blood pressure, but they're not great if we need to get stuff diffusing out of the blood into the cells, which is why when that process needs to happen, the arteries branch out into other blood vessels called capillaries. Um, and the capillaries, as you can see, have got much thinner walls. And what that helps is, again, diffusion. It allows oxygen to pass through very, very quickly into the blood, or the other way around, or carbon dioxide, whatever it may be. But things can go in and out of the blood really fast because the walls are so thick. So you often have to explain why the capillaries are so good. Thin walls, and they have a large surface area. Whenever things are small, essentially, they have a large surface area because there's so many of them. Um, right. Okay, just give me 1 second. Right. So, that's what we want to say about capillaries. Now, the third one is veins. Veins carry blood, as I say, back towards the heart. They've got the in there so that you can remember it. They carry blood into the heart. And the other features of veins, the crucial feature of veins, which again, the fact that it begins with a V should help you remember this. Because it begins with a V, it has valves. Not because it begins with a V, but it helps you remember it. So they have valves which keep the blood flowing in one direction, or prevent it from flowing backwards. Because the veins are bringing blood back towards the heart, the blood is now at a much lower pressure. It's traveling at a much less force compared to when it first got pumped before. And so we want to make sure that it keeps flowing in the same direction. And what this does is the veins keep it flowing in the same direction. Okay. And they have a larger lumen. There's other things about them that you could say. Right. Last bit of this bit before I answer that one exam question. In terms of exam questions, somebody asked if I'm going to do exam questions. I mean, probably not specifically. I'm just focusing on going through some of the content that I think is likely, but I'm trying to explain how these things are going to be asked in an exam question, or how they may be asked as I'm going along. The blood is made up of 4 different components. They are red blood cells, white blood cells, plasma, and platelets. For each of these, again, make sure you know something about them. For example, it's thinner. Red blood cells, well, let's leave red blood cells for a minute because they're the most detailed. The white blood cells, as far as this topic goes, help you fight disease. Okay, they protect you from microorganisms or pathogens. There are obviously more complicated ways in which they do that, which you do need to be aware of. But that's a different topic, really. Antibodies, all the other stuff. Right, so the platelets are for clotting blood. So they're the things that help you form a scab when you cut your skin. Okay, they form a scab when you cut your skin. Plasma basically carries everything. So plasma is the liquid that you picture when you think of blood. It carries everything, including all the red blood cells and the white blood cells and so on. They're all carried by the plasma. Red blood cells, for this topic at least, are the most important. They are as I said earlier, they carry oxygen. But how do they carry oxygen? What is it about them that allows them to carry lots of oxygen? A few different things. They don't have a nucleus. So not having a nucleus allows them to have more space to carry oxygen. So that's great. They're able to carry lots and lots of oxygen. Secondly, they have, and you want to make sure you deal with this question, they contain hemoglobin. Hemoglobin is a substance that binds with oxygen. So it keeps it in the cell. Whereas otherwise, logically, it could just diffuse straight back out again. The oxygen diffuses in, but what's stopping it from diffusing out again? Hemoglobin binds with the oxygen, keeps it there, like it sticks to it, to keep it in the cell. And it forms, and this appears on mark schemes quite a lot, oxyhemoglobin, easy enough to remember, but that's what we call the combination of oxygen and hemoglobin. When we reach the point where we want the oxygen to get out or to leave the blood, we unbind. So the oxyhemoglobin goes back to being hemoglobin and oxygen, so it unbinds with it at that particular point. And then that's the bit about white blood cells and so on. Right. So that is a little bit about the circulatory system. As I want to touch on just in this particular thing that I'm doing, just touch on a few different topics from the different sciences. Now, there was a question which said, I got this question on my mock. Explain the effect of a partly blocked artery on the human body. I mentioned that this may cause reduced blood flow and build up. Okay, fine. Let's talk about that question. Explain the effect of a partly blocked artery on the human body. So, okay, 6 marks apparently. So Paul, I mentioned that this can cause reduced blood flow. I agree. And so I'll just explain the answer that somebody gave and why they only got 1 mark. So you said it may cause reduced blood flow, which is correct. Tick. And then you said buildup of fatty deposits. Now, that's not correct, or at least that's not going to get you a mark, because it doesn't effectively what you've done is said why it might be partly blocked. So, I'm just simplifying the question a bit. The effect of a partly blocked artery on the body. Now, if you mention the buildup of fatty deposits, that's not actually explaining the effect of the blocked artery. You're saying what may have caused the blockage. So it's just a bit of a slightly, it doesn't quite answer the question. The effect on the body of this blockage isn't asking you to explain what caused the blockage. You see what I mean? Right. So that's why that doesn't quite work. However, let's explain how I would go about answering the question. Yes, if there is a partly blocked artery, there will be reduced blood flow. Fine. But what is the blood carrying? Hopefully, given that what I've just been talking about for the last 40 minutes helps you actually understand this question better. Because the whole purpose of the blood flow isn't about blood flow, it's about carrying oxygen. That's what we're trying to do. We're trying to get oxygen around our body. So if we're reducing the blood flow, aren't we reducing the amount of oxygen getting to all our cells? If we're reducing the amount of oxygen getting to all our cells, aren't we allowing the cells to do less respiration? Because the whole purpose of getting oxygen around your body is for the cells to do respiration. So if less oxygen is getting around, you're going to do less respiration. If less respiration is occurring, what is the purpose of respiration? It's to release energy. That means we're going to reduce the amount of energy being released by the cells. You see, I've already probably made this now 4 marks from that 1 mark. Then I would probably add a little bit more. If you are releasing less energy, you may, I don't know, be able to do less exercise or get tired more quickly. Or, if you want to get really advanced about this, you may end up resorting to anaerobic respiration early. I haven't really talked about this, but anaerobic respiration is when your cells have to respire without oxygen. And so that is thank you. Um, How does it unbind? Somebody quite that hemoglobin question. I don't think you need to know, but it just does. Right. Some of these things, in terms of like, I wouldn't overthink it too much because, as far as GCSE goes, there are a certain level of detail that you need to know. If you plan to continue studying this stuff beyond, then you will learn all about the detail. It's just that if you try and learn the too much detail now, you're just filling your brain up with information that you don't necessarily need to know. Like, for example, you don't need to know how it unbinds, just that it does. Okay. Can you explain required practical tools? No, I won't be doing that today. If the person said it increases blood flow, which increases oxygen levels in the cell, and less oxygen means less respiration, what do you call it? Well, it doesn't increase blood flow. A partly blocked artery quite literally reduces blood flow. So if you start with it increases blood flow, you've gone the wrong way already, right? Anaerobic respiration. You'll resort to anaerobic respiration earlier, which is the one where you're kind of running out of breath, and you're running out of breath because you're trying to get more oxygen in, and because it's not getting around your body as quickly. You might then mention, what does that produce? It produces more lactic acid, which is painful and it hurts, and so on. You're going to be in oxygen debt. There's so many things that you can go from there, but the crux of it is this bit, which is less oxygen, less respiration, less energy. And then we can explain the consequences of less energy a little bit. But that's how you go about explaining it. Differences between aerobic and anaerobic. Oh, I might come to that in a second. Also, I know you didn't go through this topic, but I understand the base of diabetes with treatments and reasons. Can you explain them slightly, please? Yeah, sure. Um, yeah, I'll do that now-ish. See what the time is. Fine, plenty of time. Differences between aerobic and anaerobic. I mean, what I'll do to answer that particular question is let me show you that table. So the differences between aerobic and anaerobic respiration are more or less in that table. Aerobic needs oxygen, anaerobic doesn't. In aerobic respiration, the products are carbon dioxide and water. So in aerobic respiration, the formula is glucose plus oxygen gives carbon dioxide plus water. These are not simple equations. Just to be clear, I'm just abbreviating the words. Whereas in anaerobic respiration, it's glucose gives lactic acid in animals, but in plants, the equation is different. It's glucose gives ethanol and carbon dioxide. Okay. So that I appreciate that's a quick summary, but the other key difference is that in aerobic respiration, we get lots of energy, and in anaerobic, we release relatively little energy. So it doesn't last for very long until you have to stop running around. Um, type 1, type 2 I'll just do this rapidly and then we will move on to some chemistry stuff. Well, simply put, it's very hard to explain type 1 and type 2 diabetes without going into a further thing. In terms of the PDF of this thing, yes, you will be able to get it because I actually made the biology slides available to everybody, but it won't just for those. There will be a way to get these. Just contact us if you need issues. But for biology ones, I'll be able to share something. For the others, possibly not. The Yeah, as I say, it's difficult to explain type 1 and type 2 diabetes without going into the bit before it, because you kind of just, I want to do this really, really fast. Right. We have a pancreas, and when you eat not lots of food, just food, and your blood glucose level gets high, okay, your pancreas releases a hormone called insulin into your blood. That insulin doesn't immediately do anything. So imagine this is this brown stuff is glucose. Don't know why brown here. Okay, loads of glucose in your blood. Right. What the insulin does is it causes other cells, specifically your liver and your muscle cells, to take the glucose out of the blood and store it. So that's what they do. The insulin is like a message for those cells to remove glucose from the blood and store it, and which means that your blood now continues merrily without the glucose that it had in it, and you have reduced your blood glucose level. That's the first thing. Still, to understand diabetes, you have to understand that bit first, which is what happens when your blood glucose level, normally in a healthy person, gets too high. So hopefully all of us here are in a situation where we have our pancreas working so that it releases insulin when your blood glucose level gets too high. And so, in answer to the question about type 1 diabetes or type 2, in type 1 diabetes, your pancreas stops producing insulin. Now, if it doesn't produce insulin, then this next step where the liver and muscle cells remove the glucose from the blood doesn't happen, which means that your blood glucose level doesn't reduce in the normal, natural way. The natural way for it to happen is for your pancreas to release insulin, and that causes your other cells to remove the glucose from the blood and bring it back. In terms of this, in terms of type 1 diabetes, because that isn't happening, the insulin bit isn't happening, the next bit doesn't happen, which is your glucose being removed. That's type 1 diabetes. And type 1 diabetes can be controlled using insulin therapy. That means we can inject the insulin into the blood that your pancreas should have been doing, but it isn't, so we can do it. We can also exercise. Exercise is a natural way of reducing your blood glucose level because it uses up glucose for respiration. And we can just avoid foods high in sugar, right? That seems like a logical thing to do. Insulin is made by a process called genetic engineering, which is a different topic, but we are able to not artificially, but in a lab, make insulin through a process that involves bacteria and DNA that produces insulin, and we do something and it produces loads of insulin, but it's in the topic of genetic engineering. Type 2 diabetes, secondly. In type 2 diabetes, your pancreas works fine. The problem is that your cells basically have stopped listening to insulin. They don't do what the insulin tells them to do. So in type 2 diabetes, whilst this step happens, as in your pancreas produces insulin, it's working fine. The second stage of this, which is where the blood glucose is removed from your blood, doesn't happen. And it's because your cells have become resistant to insulin. They stop responding to it. They don't do what it tells them to do. So that's what the issue is. Okay, that's the difference simply between type 1 and type 2 diabetes. Um, when it goes from the muscles, when it goes into the muscles, is it stored as glucose? No, it's stored as glycogen. Again, I'm not explaining absolutely every single word here, but the name is glycogen. Is insulin made using monoclonal antibodies? No, monoclonal antibodies are used in making pregnancy tests, and there are in certain cancer treatments. The insulin is made using a topic called genetic engineering. Let me answer some other questions that have popped in. Um, is this triple science and higher? Yes, is the answer to that question, but it's also combined science and higher. I mean, yeah. Bear in mind that everything is triple science in theory, right? So I think there's a common confusion here about what triple science is. Triple science is everything. Combined science is a little bit less than everything. So if I'm doing any GCSE, if you've ever seen GCSE biology, in theory, it has to be triple science because I think what you mean is it triple science only? That's a different question. Can we look into faulty valves? Eh, not particularly, but I can just show you um, where that might be relevant. So I skipped over the valves that are include a part of the heart here. So as I showed you with that crazy diagram earlier, the general direction of blood flow is like that, right? And we want this to continue going in the same direction. Now, if it doesn't go in the same direction, it could mean that the oxygen that should be going in the same direction smoothly around your body can sort of go backwards a little bit. And it doesn't mean it means that we get oxygen around our body less quickly, right? Literally, if it's going backwards or not going forwards, well, if it's going backwards, it's not going forward. So it's not going all around your body. If it's not going around your body, your cells are going to get less oxygen. Therefore, they will do less respiration. Therefore, they will release less energy. It's very similar to the blocked artery question. And this is why, again, when I teach the students I teach, we explain that this whole concept of less oxygen, less respiration, less energy can apply to quite a lot of situations. I used it in the smoking example earlier. Then we got a question that somebody asked about a blocked artery, and it was essentially the same principle: less oxygen, less respiration, less energy. The question about a faulty valve kind of is the same thing. A faulty valve will mean that we will get less oxygen around our body, which means that our cells will do less respiration, which means that we will release less energy. So it kind of all goes hand in hand, all these things. Um, Anything else? Is it an autoimmune disease? Faulty valves. If glycogen is the stored form, what's glucagon? That's a good question. Right. Okay. Tricky now. We will take a break. Somebody said we're going to have a break for 5 minutes, but yeah, I'll do that. Partly for myself than anything else. Um, so unfortunately, let me just do this now because hopefully might be helpful. There are 4 words across GCSE biology which are relatively similar. Right. And if any of you have noticed the Instagram quizzes that I've been doing, what I'll do is later on today, I will do 4 that basically have these 4 options as the answers, if that makes sense. And then you can see if you know the differences. Um, Glucose is essentially just sugar, right? It is a type of sugar. It is the product when we break down starch. What I'm telling you is everything you should know about this stuff. When we break down starch, it gets broken down into glucose by the amylase enzyme. Okay, that's what glucose is. It's just essentially sugar. Right, let me move on to no, not glucose, glycogen next. Glycogen is a stored form of glucose. So basically, when humans or animals store glucose, they store it as something called glycogen. It's basically an insoluble form of it. Um, um, there's some good questions coming in. I'll answer them gradually. Are you able to explain gas exchange one more time? No, I'm going to say no for that one. But this is being recorded. You can see it, and it's all kind of hopefully in there. So glycogen is a stored form of glucose. When animals store glucose, it needs to be an insoluble form. And so it is Yeah, that's what that is. So when I said the liver removes glucose from the blood, it stores it as glycogen. It is a form of that. If you are doing foundation level science, I don't believe you need to know what glucagon is. I'm going to say that now. Okay. But for everybody else, I'm going to mention it. Glucagon is essentially the opposite hormone to insulin. Insulin was used in this crazy diagram when our blood glucose level was too high. And what that did was helped to remove the glucose from your blood. Glucagon is a hormone that has the opposite effect. The opposite effect being it increases the amount of glucose in your blood. So if your blood glucose level is too low, such as if you've not eaten for a long time, um, your pancreas will release a hormone called glucagon, in order to raise your blood glucose level. Your blood glucose level never wants to get too high or too low. It wants to stay constant. So when it gets too high, insulin helps to bring it back down. But when it gets too low, glucagon helps to bring it back up. Glucagon is the hormone that causes the liver to basically do the opposite thing that it did before. Before, it removed the glucose from the blood and stored it. Now, what it's going to do is unstore it and put it back into the blood. So when your blood glucose level is too low, glucagon is the hormone that's released. Okay, so it's the opposite of insulin. It literally does the opposite of it. Um, okay, and then glycerol. Hopefully you know what this is, but when we break down fats or lipids, what do they get broken down into? They get broken down into fatty acids and glycerol, which is completely unrelated to everything else that I've just talked about. Okay, but they're just four words that are confusing, understandably, right? I didn't make it up. Right, so any other questions? We will take a break for 3 or 4 minutes in a second. Um, when it goes, but anybody who people are raising their hand, I don't know if that's for any particular reason, but other than there's a chat thing or a Q&A, you can just ask things in there. Um, why does there have to be a specific location for an artificial pacemaker? Well, I think it's because the actual pacemaker is in a specific location. Your pacemaker cells are in, I want to say the right atrium. Where they go. And so the artificial one has to be in a specific location as well. Glucagon done. Um, someone has written something long. Antibiotics treat food poisoning because they kill salmonella bacteria. Some antibiotics work because they damage the bacteria cell. Um, okay, I'm not answering that question. Right, what we're going to do is we are going to I'll take a break for a few minutes, and then I'm going to come back with some chemistry, specifically a topic called electrolysis. Now, somebody asked me about topic four. Topic four in AQA, just to be clear, I have to be very clear about exam boards because the order of topics is different. So when I say topic four, I'm talking about AQA. So if you're doing a different exam board, it's still in there. It's just not necessarily topic four. Um, Contains various things, including neutralization, acids and alkalis, strong and weak acids, reactions of acids with metals and stuff. Um, and then it also shifts onto displacement reactions and electrolysis. And that's the bit I'm just going to talk about. Because again, I think that a lot of people don't do this bit very well. And it is quite complicated. And so I'm going to talk a little bit about electrolysis, but we will do that at 5 minutes past 4. Will I go over moles? Yeah, but briefly. I might just have to stick to chemistry time-wise. Um, Fuel cells I'm going to do in the session that I do tomorrow because it shifts into triple science a little bit. That's what fuel cells is. The electrolysis bit of it doesn't. I understand it is kind of similar. Right, I'll be back in 5 minutes. Um, right. Okay. A couple of questions. Will people who aren't usually part of your classes have access to the uploaded videos? Yes, they're just going to be on YouTube. The YouTube is, um, I mean, I don't know write the first bit, do I? Slash Step to Success UK. I'm sure you know the first bit of YouTube. Okay, that's what it is, right? And you should be able to find it. It should be on there automatically after this session. If it's not, I will put it up there tomorrow morning. Just to make sure, because I've got a separate recording of it. Um, bum, bum, bum. Could you explain balanced equations? We might come across one or two, yes. Titration calculations. The titration calculation I'm not going to do today. I've specifically allocated tomorrow's session to do some triple science specific stuff. Okay, so things like titrations, things like fuel cells, things like I don't know, monoclonal antibodies, whatever it might be. I'll be specific to that. I'll do tomorrow. Just that it's more relevant to everybody today, and then I can tell everybody now. Um, I had a question to identify the compound of the electrode or the electrolyte into conducting experiment in this way. It depends on the question that you're. I'd have to look at the question specifically. I wouldn't say there's a general method. It just depends on what the question says. Um, Protein synthesis again. That is something I'm going to do. I can do in the triple bits tomorrow. I don't know what you mean by types of reactions exactly. Right. So let me focus on a specific type of reaction, which is a displacement reaction. Firstly, now in a displacement reaction, the idea is that a less reactive I'll find the link and I'll send it to everybody in the chat in a minute of the YouTube at the end. A less reactive metal can displace a more reactive metal can displace a less reactive metal. There is something called a reactivity series, which has things like potassium, sodium, lithium, and then it has like carbon somewhere, then it has like copper at the bottom, and then it has some other stuff like iron and zinc around here somewhere. I can't remember the order exactly. But there is a reactivity series of metals. Now, if, for example, I had some copper oxide, okay, and I found copper oxide in like a rock, and this is what happened with a lot of metals. You find them already reacted with something in the air. So it's difficult sometimes to find only copper because if I've got copper just lying around, it will usually react with the oxygen in the air and become copper oxide or something else. And now that's a problem for us because copper oxide doesn't conduct electricity, but copper does. And the thing that we want copper, or we want something to be able to conduct electricity for our wires. I can't put copper oxide in our wires. I'd have to put copper in the wires so that it can conduct electricity. So often what we're trying to do, and this is where this whole topic begins really, is make a change to this chemical changes, that allows us to get the copper separated out and the oxygen fine separated out, but the copper is the important bit. One way to do that is to react the copper oxide with a element that's more reactive than it in this reactivity series. Conveniently, there are lots of things more reactive than copper because copper is right near the bottom. If I react copper with, let's say, iron, then what happens is that the iron displaces the copper. Effectively, what I like to think of that is that it kicks it out. Um, so that becomes iron oxide. It takes its place, essentially, displaces it, and the copper gets kicked out and separated out. So we have got the copper separated out. Um, so that's precisely that, okay? Why is it always copper in the questions that they ask you? Partly because copper is so unreactive. And so, as I'm about to explain, actually, the fact that copper is near the bottom means that there's lots of things above it that we can use to displace it with. So we need something more reactive than copper. Um, Whereas something like lithium oxide, yes, they can ask you about it, but the point is that it's less common to have to do this because it is harder to find um, an element that's more reactive than lithium to kick it out. You need something more reactive than an element to be able to displace it from its compound. And it's harder to do that with something like lithium because there's fewer options available, like above it. Whereas with copper, you've got all of these options. So you're more likely to get questions on it because of the fact it's copper. Um, can you give us questions on the things we learn? I'm not quite sure what that means. Will I need to remember the reactivity series for my mocks? Well, I mean, no, I don't think you need to learn the reactivity series as such. But the I would say that in principle, you should be aware that things like potassium, sodium, lithium are near the top. Things like copper are near the bottom because they're very unreactive, and carbon and stuff is in the middle. I think that sort of broad understanding of things should be fine. And bear in mind that the first three elements are just in group one in reverse. It goes lithium, sodium, potassium is just in reverse. So you kind of already got some of it already there. Um, can we do some balancing equations and atom economy? Um, I'm not going to do atom economies yet, but we'll talk about it tomorrow if you want to join tomorrow for the triple science. And I'll do balance. I mean, I'm not going to spend the reason I'm not going to do balancing equations. I'll just tell you why. Is because look, in the grand scheme of things, you're not going to get a big question on balancing equations, right? It is a smallish aspect of the syllabus. It might be worth a mark. Don't stress over it too much. Electrolysis, I'm getting to. Right. So with certain compounds like copper oxide, it is very easy to find an element that's more reactive than it to displace it in this method. So iron is an example. I could have used zinc. I could have used carbon. I could have used lithium. There's loads of stuff above it that can kick it out. However, as we get higher up on the on the reactivity series, like to sodium, for example, we can't really use this method because there aren't as many readily available elements that are more reactive than sodium that I can react sodium oxide with, for example, to kick out the sodium. So it doesn't quite work as we get higher up, which is why something called electrolysis exists. So I'm going to do two examples of two electrolyses that you want to be familiar with. The first one is the electrolysis of aluminium oxide, and it is molten aluminium oxide. I want to explain that word in a minute. But aluminium is quite a reactive metal, which means that if I find some aluminium oxide and I only want the aluminium out of it, then it's quite difficult to use this displacement reaction method because that requires an element that's more reactive than aluminium to react it with. So it's not as easy to get hold of these elements that are quite that are very reactive, like because it's just hard to find them. At least it's hard to find them unreacted because that's what you need. So we can't really use a displacement reaction to separate or to get the aluminium out of aluminium oxide. That's where we use electrolysis. Right. Let me just go through a little bit of a summary on this. Aluminium is in group 3 of the periodic table. Oxygen is in group 6 of the periodic table. On the basis that it's in group 3, it means that aluminium atoms lose 3 electrons when they need to get a full outer shell, which means that the aluminium is a 3 plus ion. Quite important. The oxygen atoms, because they've got 6 outer shell electrons, will gain 2 to get a full outer shell, which means that this is an O2 minus ion. Al3 plus O2 minus. That is the name of the ions. You can sort of get that if you get given the formula. Aluminium oxide is Al2O3. Sorry, Al. Yeah, it is. Sorry, Al2O3. And it from those numbers you can often figure out what the charges on the ions are because that 3 is on top of the 3 there, and the 2 is on top of the O there. So it can be done. But anyway, I'm telling you how to do it from the periodic table, which is better. So the next bit is in order to do any electrolysis, we cannot do it with a solid compound. Okay, so if it was a solid, then it would look like this. We'd have the positive and negative ions, the aluminium and the oxygen ions, sort of in this rigid structure, however we describe this, um, where the ions cannot move around. We therefore have to, in order to allow them to move around, either melt the compound or mix it with water, which means in solution. Okay, so we have to do one of those two things. If it dissolves in water, then we use the in solution one. If not, we have to melt it. Aluminium oxide does not melt. Sorry, does not dissolve in water. Therefore, we have to melt it. So that's why we're going to use molten aluminium oxide for this. Can't write that. For this electrolysis, right? And then what we're going to do is melt it, and we're going to put it into a beaker. We're going to put these positive electrodes. Somebody asked about inert electrodes a little while ago. Inert just means they are unreactive. That's all it means. It just means that we don't want these electrodes, preferably, to react with things. Sometimes they do because they're made of carbon. But what we do is we connect a battery, physics, circuit symbol, battery, to each side here, which means we get a positive side and a negative side. The positive electrode is called the anode. The negative one is called the cathode. And what we do is we melt our aluminium oxide, which, as you saw from that diagram earlier, was made of positive and negative ions. And those positive and negative ions, because they're now in sort of a liquid form, can move around. That's the crucial bit now. The point of all of this, the point of melting this, is that we want these ions to move around. And by getting them to move around, we can hopefully separate them. Remember, what we're trying to do is separate aluminium and oxygen, get it separated out. And what happens here, because we've got a positive and a negative side, and hopefully you know the opposites attract, the positive aluminium ions attract to the negative electrode, and the negative oxygen ions, or oxide ions, attract to the positive electrode. And therefore, I mean, we've sort of done it because all we get now is aluminium, pure aluminium, not aluminium oxide, but pure aluminium on one side, because that's where all of the aluminium ions have gone. And we get oxygen gas being given off on the positive side. Okay. I will point out that once you, the solution at the bottom is called an electrolyte. So that's what's happened here. We've got two products from this electrolysis, aluminium and oxygen. However, there are these things called half equations, which are stressful. The half equation for aluminium goes like this. This is how I recommend you do half equations. And also, when I give you my the YouTube thing, which I do now, there is a specific electrolysis video on there, which, if I do say so myself, is really good. Which I would recommend watching. Okay. Let me see if I can find my link. I think that link should take you to it, hopefully. Hopefully it does. If not, if you go on our website as well, I think there's a link to it somewhere on the top. Anyway, let me continue. But yeah, if you are really struggling with electrolysis, and obviously this brief session is not going to necessarily make you an expert on it if you are struggling, then that would be a good way to do it. So here is an aluminium 3+ ion. What we need to understand is what actually happens here. We started off with an ion and we ended up with the original atom. That's what we're trying to understand. So we started off with Al3+, we ended up with aluminium. Okay, that's what we've ended up with. Now, how does that happen? This tests your understanding of ionic bonding properly, because if you think about how an aluminium atom became an aluminium ion, well, how did it do that? How did it become positively charged? It must have lost 3 electrons to become an Al3+ ion. If it's going backwards now, if it's going from the iron back to what it originally was, surely it needs to gain 3 electrons to go backwards. Okay. And so if we add 3 electrons to this, we will go from an Al3 plus iron to back to the original aluminium. Because again, how did it become an Al3 plus iron? It lost 3 electrons. Now it needs to gain those back. And that is the half equation. Simple way to remember half equations is that initially, all you need to write, this thing here should be the same as this thing here, both the charge and the number initially. It doesn't always work like that. But to start with, I always write that down. So whatever the charge is, 3 plus, put plus 3 electrons after it. If it's a minus, put minus that many electrons, although that might change, as we'll see in a minute. So what's happening to the oxygen? This is the oxygen ion. What happened to it? Well, it turned back into the oxygen atom. This is, again, if you don't quite understand this, it's mainly because your understanding of ionic bonding is a little bit weaker, and that needs to be sort of rectified. So an O2 minus ion, what does it go back to being? It goes back to being oxygen during this process. We are an ion, we're going back to being the atom. So to do that, again, the first thing I always write when I'm trying to do a half equation is whatever is here, I put after it. So if that's a minus, I put a minus. If it's a 2, I put 2 electrons. That's what I always do first. Okay. And that isn't unfortunately complete, just because when you have a gas produced in an electrolysis, like oxygen, it comes off as what we describe as a diatomic molecule, but the point is it has a 2 after it. So that isn't actually what we get at the end of it. We get O2 at the end of it. Because we put a 2 there, what we have to do is we have to double the number of ions. This is essentially balancing equations. And we have to double the amount of electrons. So the -2 becomes a -4. That is the half equation. Okay. And again, the reason why people get confused about these half equations is because often when you're presented with this, or when you see this written down, they write it like this, which doesn't make any sense. But just so you understand where that has come from, the -4 electrons have just been added to the other side, like in a maths equation. So if I've got a negative on one side, I can add it to the other side. They just do that because presentationally, in chemistry, we don't really subtract things. You don't have this minus this, do you? It's always that plus that in equations, like photosynthesis or whatever. So this -4 has just been added to the other side. So presentationally, it's sometimes written like that. And I think that's part of the reason why people don't understand it, because that to me doesn't make any sense. But that does make sense because I understand where it came from. I understand that I had an O2 minus ion. It had previously gained 2 electrons to become an ion. Now it's lost those electrons, and then you just balance everything. So that is the half equation for that. As I say, I highly recommend on that YouTube somewhere, there is something called an electrolysis workshop, whatever that means, in which I did try to explain this all from the beginning, and I go through these specific examples, including this one again. So the 2 half equations are there. Okay. For this electrolysis, okay, for this one. I will also mention one more word involved with this electrolysis specifically, which I always see questions on, and it's to do with something called cryolite. Cryolite is a substance that's added to the aluminium oxide to lower its melting point. Because in order to melt aluminium oxide, in order to melt any ionic compound, it gets very expensive. It requires a lot of heat because ionic compounds have strong electrostatic forces of attraction. So it takes a lot of energy to melt them. So it would be so expensive to melt a lot of aluminium oxide to do this electrolysis, it would almost not be worth it to get the aluminium out of it. So adding this thing called cryolite helps to lower the melting point of the aluminium oxide, making the whole process use less energy, and therefore it becomes cheaper. So just so you know, in case you ever see the word, cryolite, that's what it relates to. Would you get the mark for both? Are we allowed to present it the way your first time? Well, so it's a good question. If they ask you to just write out the half equation with no other information, if I give you a blank line and say write out the half equation, either of these two will be correct. it doesn't matter because they are actually both the same thing. The issue that happens here, though, is that sometimes what they do is they give you part of the half equation and ask you to complete it. So in this case, they might say something like this: complete the half equation. Like that, right? So you have to understand that sometimes they will have already done the rearranging bit and then ask you to just complete it. So that means they've already written it as they want to write it. So you have to just understand how it happens. So if you have to write it from scratch, then yes, fine, just write it as either of these two is fine. You'll get the marks. But sometimes it's half written out for you already, as you've probably seen. So then you have to just understand what's happened. That's why I'm trying to explain it. Right, we will do one more electrolysis, a more complicated electrolysis, partly, which is the electrolysis. And this is one of the required practicals, by the way, for those somebody mentioned it earlier, which is the electrolysis of sodium chloride solution. Okay. First things, the previous electrolysis that we did, yes, this is all relevant to everybody, combined and triple, yes. The previous thing that we did was aluminium oxide. It didn't have the word solution written after it. In fact, it had the word molten at the front. As I mentioned earlier, there are two ways to do an electrolysis. You can either melt the thing you're trying to separate, or you can dissolve it in water. Okay? And that's where the word solution comes in. The word solution means this stuff has been dissolved in water. And that creates a complication as far as electrolysis goes. When it came to this previous electrolysis, it was quite straightforward, really, what happened. The positive things went one way, the aluminium, the negative things, the oxygen, went the other way. Relatively simple. Okay. But now it gets more complicated because sodium chloride solution. Firstly, let's talk about sodium chloride. Sodium chloride is made up of sodium ions combined with chloride ions. Their ions are Na plus and Cl minus. You should technically know that because sodium's in group 1, chlorine's in group 7. From that only, you should know what their charges on their ions would be. The problem is the solution. I mean, are you just asking me questions, just directly copied and pasted from them? Cryolite contains Na plus ions as well as Al3 plus ions, so that's one reason why sodium's not. I'm coming to that sort of in this concept. It's to do with reactivity. I want to explain it now. So, sodium chloride is made up of Na plus and Cl minus ions. Solution means that there's water there, which there wasn't in the previous electrolysis, right? We didn't have water here. We didn't have a solution. We just had aluminium and oxygen. The solution, when we run, is water. And when we run electricity through this, as we're about to do, it forms H plus and OH minus ions. Okay, so that solution is now going to have additional ions. It's going to create additional ions inside our electrolysis. So H plus and OH minus is the water, right? H2O. So if I've got sodium chloride solution there, and sometimes you have to do what I'm doing, by the way, in terms of actually drawing this electrolysis out. It's not difficult, but do make sure you can do it. So that is my electrolysis. This is my sodium chloride solution. In sodium chloride solution, there are all of these ions now. And this is what makes it different from the previous question. In the previous question, we only had this positive ion and this negative ion. Whereas now we have got two positive ions, Na plus and H plus, and two negative ions, Cl minus and OH minus. Do you see what I mean? It's only because of the water that has happened. The water in there means that we've got these two additional ions, positive and negative ones. So whilst it was obvious what happens in this electrolysis, the positive ones go one way, negative ones go the other. It is not as straightforward for this electrolysis because what you must understand is that out of the two positive ions, only one of them will go and be produced at the negative electrode. And of the two negative ions, only one of them will go and be produced at the positive electrode. That wasn't an issue that we had to deal with in the previous one because there was only one of each. The positive ions went one way, negative the other. Now that we've got one of each, sorry, I'll keep elaborating this point. Now that we've got one of each, only one of them goes. Okay. And you need to understand how you know which one goes. Let me explain. For the positive ions, there is a rule to determine which of the two will go to the negative electrode. And the rule is the following. The less reactive of the two ions goes to the negative electrode. Just put the electrode. So of sodium and hydrogen, the less reactive one is hydrogen. And that is where that reactivity series and having a rough understanding of what it looks like. I don't know where I put it now. A rough understanding, I think I got rid of it, of what it is helpful because you will know that sodium is really high up on the reactivity series and hydrogen is now right near the bottom. Hydrogen, copper right at the bottom. So you know that sodium is the more reactive than hydrogen. And the fact that it's in group one should tell you as well, because group one metals are very reactive. So sodium is more reactive than hydrogen. So the less reactive of these two is hydrogen. So it's the hydrogen ions that will go there and be produced at the negative electrode. And we're going to end up with hydrogen gas coming off here at this point. Okay, that's what's going to happen. The sodium ions will stay in the solution. It's the hydrogen ions, because they are less reactive, they will go to the other negative electrode. That's the rule for the positive ions. Unfortunately, the rule for the negative ions is different again, which makes the whole thing painful. Is the presence of H plus ions a result of sodium chloride being aqueous? So there's water. It's not that sodium chloride is aqueous, just to be clear. Sodium chloride is it's salt, literally salt. That's table salt. That's what sodium chloride is. The salt that you will have in your kitchen at the moment. So it itself is not aqueous. It's just that to do the electrolysis, what we do, the easiest way to do the electrolysis is to make it aqueous by mixing it with water. That's what we have done. We have added the water. All of this is the electrolysis of salt and water. That's all this is. That's why it's so easy to show you in class. We literally the electrolysis of salt mixed with water. So the salt is the compound, sodium chloride. In order to make it conduct electricity that we need to happen for this electrolysis, we need to either melt it or mix it with water. It is much easier to mix it with water because melting sodium chloride would take a lot of energy. So it's just easier to put it into water. So, but that creates this issue of H plus and OH minus ions. Hopefully that makes sense. Right, of the two negative ions, the rule is a little bit convoluted. How do we know it's less reactive? I did explain that because there's a reactivity series that tells you. The rule for the negative ions is that of the two, remember, one of them will always be an OH minus ion because that's why we've got two in the first place, right? Because we've added water in. We've only got two negative ions because we've added water. So one of them will always be an OH minus ion. The rule is for the negative ions that the OH minus ion always goes. I'm using simplified terminology here. The correct word is discharged at the electrode. I'm just going to stick with it. The OH minus ion always goes to the electrode. And then this is the convoluted bit, unless the other ion is a group 7 ion. Okay, so unless the other ion is a group 7 ion, the OH minus ion always goes. So in this case, the OH minus ion always goes unless the other one is a group 7 ion. And guess what? In this example, the other one is a group 7 ion. Chlorine is in group 7. So that means that it's the chlorine that gets produced at the positive electrode. So in this case, the hydrogen ions go there, and the chlorine ions go to the positive electrode, leaving behind the Na plus and the OH minus ions inside the solution. And we end up with chlorine gas coming off on that electrode and hydrogen coming off at the other electrode. And if you have been shown this practical in school, this is where you may have been shown because it's a sensible time to do it. Um, the tests for chlorine and for hydrogen. This is the point at which you might have been shown that hydrogen burns with a squeaky pop because it's there. We just get it off. We put a test tube over here and we collect the hydrogen gas. You may have been shown that chlorine bleaches damp litmus paper, and you may have been shown that because chlorine has just come off here. So it is a good, sensible time to show you that, given that it's in the syllabus. What is a halide? A halide is the name given to a group 7 ion. That's what's called halide ion because it's a halogen. It comes from the word halogen, group 7, halide. So that's why is the case only group 7? Don't worry about that. It just is. So the in this electrolysis, we have got sodium, hydrogen, and chlorine. What I need to do now quickly is the half equations for this, because the half equations we only do for the things that come off at the electrode. So hydrogen and chlorine, they're the two half equations we need to do. Hydrogen is a H plus ion. What has happened to the H plus ion? It has turned back into hydrogen. That's what's happening. How does a 1 plus ion turn back into the original atom? It gains one electron. Remember, if there's a 1 plus here, put a 1 plus there to start with. Then, unfortunately, just like with the, what did we see earlier? Oxygen in the previous one, because oxygen comes off as a 2, hydrogen does as well, it means that you have to double everything earlier on in the equation. So I always recommend when you're doing half equations, try and understand half equations, do it like this first, because I feel it makes more sense. They go, okay, I've got H plus ion, it gains an electron, it goes back to being H. But it's not H, it's H2. So everything needs to double. Similarly, for chlorine, chlorine is a Cl minus ion. What does it do? It turns back into chlorine. How does it turn back into chlorine? It loses an electron. Again, whatever this is what this should be to start with. Whatever that plus is, should be what that is. Because again, a negative ion must have gained an electron, and now it will lose that electron to go back to being chlorine. But once again, chlorine is a gas that comes off as a 2. So you now double everything before it, and you get your half equations. Once again, sometimes that subtraction can be moved to the other side. But if you understand it up to now, then that last bit should be easy. Why for oxygen was the amount of electrons 4? If we need to double it, why wasn't it 2 electrons? Because we doubled an existing 2. So let me just go back to this. So this was what I originally wrote down. I wrote down that I've got an O2 minus ion, and it goes back to being oxygen. How does an O2 minus ion reverse itself? Well, previously it gained 2 electrons to become an O2 minus. Now it's going to lose those 2 electrons to go back to being that. So that's why I write first, just to make everything correct, because that is true. Now, an O2 minus ion loses 2 electrons, goes back to being the original atom. Unfortunately, the original atom is doubled, which means that everything else has to double. That's why I put a 2 there. That's why I double the 2 that I wrote before to make it a 4. So that's why the oxygen has a 4 and the hydrogen doesn't, because hydrogen is a 1 plus ion that's doubled to a 2, essentially. Hopefully that all makes some sense. There is a tiny bit more for this electrolysis. So many things. Which is that during this electrolysis, what has happened? The hydrogen has gone to the electrode. The chlorine has gone to the other electrode. What's left behind in the solution? We have got these Na plus ions and these OH minus ions, which join together to form NaOH, sodium hydroxide. So often you get asked that there's or mentioned that there's 3 products in this electrolysis, and there are. The 3 products are chlorine at one electrode, hydrogen at the other electrode, and sodium hydroxide that's left in the solution at the end. That's that, right? Okay, and then lastly, I'm just going to mention something called oil rig for this bit. Um, oil rig is, well, it stands for oxidation is loss and reduction is gain of electrons. That is important. I should make sure. Has anybody asked me anything else? Um, oh, um, Okay, that cryolite question. So you mentioned the cryolite contains Na plus ions. So in the electrolysis of aluminium oxide, we've got Al3 plus and O2 minus. You might get this kind of question where they add a bit more detail to the cryolite situation. They say cryolite contains Na plus ions as well as Al3 plus ions. So cryolite is a bit more aluminium, but it contains some sodium as well. Suggest one reason why sodium is not the product for electrolysis. Well, you should actually know the answer to that question now, because what happened in the previous electrolysis? What was the rule about the positive ions? That the less reactive ion is the one that gets produced at the electrode if there are two positive ions. We're being told in this question that there are two positive ions here. Of those two, we're being told that aluminium is the one that is the product. Why is sodium not the product? It must be because aluminium is less reactive than sodium, or because sodium is more reactive than aluminium. Either way, the less reactive one of the two is the one that gets produced. That's the answer to that question. Um, I'm not going to get to physics, forces, AQA. We can deal with that if you're here with me later on today. We can do a bit of that then. Was there anything else in electrolysis? Yeah, oil rig, I was about to talk about. Oil rig is, or it stands for oxidation is loss, reduction is gain, but very importantly, of electrons. It's very important that you understand that the oil rig is referring to electrons. This little acronym that's very easy to remember. And it's like, oh, lovely, oil, oxidation is loss, reduction is gain. What does it mean? It's talking about electrons. So in every electrolysis, I've only done two, but in every electrolysis half equation that I've done, if you notice, we just go back to them. If we look at the first lines here, in all of these half equations, one of the substances gained electrons and one of them lost electrons. If we look at oil rig, then we know which one's happening here. Because oxidation is the loss of electrons, reduction is the gain of electrons. So what's happening here can be described as reduction because we are gaining electrons. Here, what's happening? Electrons are being lost. So we can describe that as oxidation. Again, sometimes you get asked these questions like, why is this process oxidation or why is this process reduction? And you can just use oil rig to explain it. But I'm just showing you in the half equation that in the two equations, something got oxidized, something got reduced. So oxidation was loss of electrons, reduction is gain of electrons. Similarly, if we go back to these two half equations, something gained electrons, so that is reduction. Something lost electrons, so that was oxidation. You occasionally get asked that to be able to deal with it. Right. Is there much else that I wanted to focus on? So electrolysis, I've done. Right, just for the last few minutes. Again, I recommend that on that YouTube thing, find the electrolysis thing I did. I think it works. It's really good. So just on this, on the beginnings of quantitative chemistry, very much the beginnings. They go into some detail, as you all know. There is a formula, which is moles is mass over m. I think people just don't get moles. So I'm going to give you a tiny, tiny little intro on it, and then that'll be it for today. But the whole point of moles is that it's a unit of measurement in chemistry. It is a unit of measurement to measure the amount of a chemical that I've got. Oops. There are two ways. If I've got some chemical in my hand, okay, then there are two ways for me to say how much I've got. I could measure it in grams and literally put it on a weighing scale, and it'll tell me it's 30 grams or whatever. Or I could use moles as a unit of measurement. Just like, you know how you can measure distance in like miles and kilometers, and the same way you can measure temperature in Fahrenheit or Celsius. In the same way, we can measure the amount of a chemical in grams or in moles. And just like with those other examples that I just said, like miles and kilometers or whatever I said, degrees Celsius and Fahrenheit, right? There is a way to convert from one to the other. And that's what this formula is, okay? All it's doing is converting an amount in grams into moles It is just a different unit of measuring the same thing. There are benefits of using moles that I won't go into, but really, just to really simplify it in your mind, moles are just a unit of measurement for an amount of a chemical, which we can use instead of saying I've got 30 grams of it. Let me give you one example. And I guess we need to mention MR here. The MR is relative formula mass, which is the sum of the masses of the atoms in the compound. For example, if I had CO2 and I said, what is the relative formula mass of CO2? Probably in the question, it'll tell you that carbon has a mass number of 12 and oxygen has a mass number of 16 from the periodic table. If I wanted the relative formula mass of carbon dioxide, I would do 12 plus oxygen, which is 16, but there's two of them. So it's two lots of 16. 12 plus two lots of 16 is 12 plus 32, which is 44. That is the MR of carbon dioxide. Now, let me give you one example. If I said to you, how many moles is 88 grams of carbon dioxide? Let's say I've got 88 grams of carbon dioxide, although that's a tricky thing to explain. But 88 grams of carbon dioxide. If I said to you how many moles is that, then remember we could just convert grams to moles by taking the mass in grams and dividing by the MR of the compound. The mass in grams in the question is 88. The MR I just worked out a minute ago of carbon dioxide is 44, which means that that is 2 moles. 2 moles of carbon dioxide is the same as having 88 grams of carbon dioxide. So that is essentially how we turn grams into moles. Now, firstly, that's important that you just understand that there is a thing there in terms of all it is a conversion from one to the other. Right. A hydrocarbon fuel is completely burned in excess oxygen. I'm not answering that question. I'm not going to be able to do any more questions now because I have other classes to teach at 5 p.m. unfortunately. So the that's it for now. So if you want to do more, just so you know, tomorrow morning, sadly at 9 a.m. because I have no other time, I'm going to do some foundation level maths stuff that is still relevant if you're doing high level. Remember, half of a foundation level paper is the first half of a higher level paper, so it's not like they're completely different things. So feel free to join at 9ish. And then I think same time again at 3 o'clock, I'll be doing some more science, but I'll be focusing slightly more on the triple science side of things, like harder stuff like titrations and monoclonal antibodies and space, maybe in physics and whatever else I can think of. The pre-recorded maths. So the link is the same, correct? Yes, I should say that. The link is the same for tomorrow, exactly the same place. The maths that I did this morning, I will put on YouTube later on tonight. Okay, so I think it's not automatically ended up there because I didn't press a button. Yes, so today's one will end up there later on. The session that I've just done now, as soon as I stop this, hopefully will end up on YouTube shortly after. It takes a little while, but it's not long. Hopefully it'll be there, and then tomorrow's will hopefully go on. It's just this morning I messed up, but it's recorded. I just need to upload it if that makes sense. So that's it. Hopefully that was helpful for everybody, and I'll see you tomorrow.